Welcome to the wonderful world of gametes. Gametes are haploid sex cells consisting of an egg and sperm, which fuses together to form a diploid zygote. Without sexual reproduction, there would be very low genetic variation amongst individuals. Ah, the science of life.
The ovum is the female gamete, capable of giving rise to a new individual in every cell it contains. No other cell has this amazing capability. Activation of the ovum and all of its potential occurs when the sperm and egg fuse. However, sperm isn't always required. Parthenogenesis is the development of an ovum into an embryo without a sperm cell. Many animal species are capable of parthenogenesis, including lizards, birds, snakes, sharks, aphids, daphnia, rotifers, and nematodes. Typically spherical or ovoid, size varies across species, with the typical human ovum being 0.1 millimeters in size. Fish and frog eggs are around 1 to 2 millimeters, and reptile and bird eggs capable of being many centimeters in length. Female gametes are giant single cells full of nutrients needed to sustain the organism until it can begin taking in its own. The cell's cytoplasm is very important because it contains the nutrient reserves for the embryo. The ovum's cytoplasm is also known as its ooplasm, or yolk. The ooplasm contains the nucleus and nucleolus. It is made up of two parts, the spongioplasm, formative yolk, and hyaloplasm, nutritive yolk. The hyaloplasm is rich in lipids, proteins, and polysaccharides, and is sometimes contained by a membrane. Ooplasm contains ribosomes, tRNA, mRNA, which remains dormant until fertilized, and morphinogenic factors, which are molecules that direct the differentiation of cells. Some species' eggs also contain protective chemicals such as ultraviolet filters and DNA repair enzymes to protect the embryo from the sun, antibodies, and distasteful chemicals to. The egg coat is an extracellular matrix made up mostly by glycoprotein molecules. Many mammals have a plasma membrane for their major coat, called the zona pellucida, which immediately surrounds the egg. In non-mammalian eggs, the major coat is called the vitelline layer, which is made up of protein fibers and fiber receptors for sperm binding. The vitelline layer aids in protection and only allowing same species or closely related species sperm in. The jelly mass, shell, membranes, or albumin is not part of the ovum. It is secreted around the ovum as it passes down the oviduct. The nuclear components of the ovum are the same as the spermatozoa. Spermatozoa, highly specialized for speed and efficiency in fertilization. It doesn't have any unnecessary cytoplasmic organelles like ribosomes, endoplasmic reticulum, or the Golgi apparatus. Sperm cells are composed of two distinct regions enclosed by a single plasma, making up one sperm cell, the tail and the head. The head of the sperm contains DNA in the nucleus, which is packed extremely tight by highly positively charged proteins called protoamines. On the interior part of the head is the acrosome, a specialized secretory vesicle that contains hydrolytic enzymes that help the sperm penetrate the egg's outer coat. The tail of a sperm is a long flagellum whose central axoneme emanates from a basal body just below the nucleus. The axoneme consists of two central singlet microtubules surrounded by 9 evenly spaced microtubule doublets, and some have another 9 outer dense fibers composed of keratin. Flagellar movement is driven by dynein motor proteins fueled by ATP to slide the microtubules past one another. Sperm contain many specialized mitochondria in the midpiece, which are strategically placed around the flagella for the most power. Semen contains fructose as the main energy source for the mitochondria to produce ATP.
Spermatogenesis is the formation of sperm cells. Before meiosis 1 takes place, the spermatogonium is replicated during interphase so that its two end single chromosomes become two end sister chromatids. The spermatogonium then divides by mitosis, creating two daughter cells. One daughter cell becomes a primary spermatocyte, while the other daughter cell remains a spermatogonium. Next, the primary spermatocyte divides by meiosis to produce secondary spermatocytes, which now have end sister chromatids. The secondary spermatocytes divide again, which yields a number of 4 spermatids. These spermatids later mature into sperm cells, each having end single chromosomes and are thus haploid. Genesis is the formation of egg cells. The primary oocyte is replicated during interphase so that it has two end sister chromatids. Meiosis 1 takes place, and the primary oocyte is divided into secondary oocyte and a polar body, each retaining end sister chromatids. After meiosis 2, 3 more polar bodies are produced and 1 ovum is created. The polar bodies eventually disintegrate while the ovum matures with end single chromosomes.
The zona pellucida is the porous extracellular matrix surrounding the vitelline layer of the ovum. It plays a critical role in binding spermatozoa to egg and inducing the release of acrosomal contents. It is involved in the complex process that restricts interspecies fertilization. This picture shows the surface, which in mice is composed of 3 major sulfated glycoproteins that are non-covalently linked. However, humans, being higher species, have a 4th glycoprotein. The stages of fertilization. First, the sperm head binds to the zona pellucida with help from the fertilin disintegrin domain that interacts with the integrin of the egg. Second, exocytosis occurs, which is when the acrosomal contents are released and reaction takes place with the trypsin-like enzyme. Third, the sperm penetrates the zona pellucida and the vitelline layer of the egg. Fourth, the fusion of the egg and sperm plasma membranes. And finally, the sperm nucleus enters the egg cytoplasm. Some important proteins and enzymes involved in fertilization are fertilin, which is a protein found on the sperm surface and is composed of alpha and beta fertilin subunits. It mediates the first binding step with an interaction between its disintegrin domain with the egg's integrin unit. Secondly, there is the epidermal DE protein, which is thought to be a part of the subsequent step following the sperm-egg binding that leads to fusion. However, not much is known other than the fact that there is no disintegrin domain involved. Lastly, there is trypsin-like enzymes, which are stored in the acrosome, and they are released after the sperm binds to the egg to disintegrate and penetrate the vitelline layer. The proposed mechanism involves a germline disc on the egg, which is a spot on the egg with a different amino acid sequence or composition than the rest of the egg. The sperm uses this information to know where to bind to on the egg for the easiest entry.
Welcome to the wonderful
Powered by Video.